once again and welcome. So for today, it is just a continuation of our previous discussion relating to neurons and neuronal. So as we have said previously, there are two major types of cells which are found in the nervous systems. We have the glial cells, which provide physical and metabolic support to neurons, including neuronal insulation and communication, and nutrients and waste transport. Neurons act as interconnected information processor. So, as we know that we have these different parts of the neurons. So, we have the dendrites, which receives information. Then we have the soma, or the cell body, the axon helix, the axon, the myelin sheaths, the nodes of Lanier, and the terminal button. So, neuron structure. Okay, so we will be talking about synapses, neuronal neurotransmitters, and receptors. Okay, as we know that neurons do not touch each other, but where the neuron does come close to another neuron, a synapse is formed between the two. So, synapse is the place between two neurons and the site of communication, while neurotransmitters are chemical messengers that bind to receptors on the next neuron. Then receptors are proteins on the cell surface where neurotransmitters attach. They vary in shapes, with different shapes it matching different neurotransmitters. So a synapse, as we said, is a combination of presynaptic endings, which contains the neurotransmitters or the chemical messengers. We have the synaptic cleft, which is the gap between the two neurons, and then the postsynaptic endings, which contains the site of receptors, where the molecules which receive signals for a cell. So, synapses can either be chemical or electrical and are essential to the functioning of the neuronal or the neural activity. Neuroscientists understands that synapses plays a vital role in a variety of cognitive functions, including learning and memory formation. So, electrical synapses are different from chemical synapses, as there is a different or a direct physical connection between the presynaptic and the postsynaptic neuron. This connection takes the form of something called a gap junction, which is essentially a channel which allows ions to flow directly from the presynaptic cells to the postsynaptic cell. So gap junctions contain paired channels in the membranes of the pre- and the postsynaptic neurons, forming pores. And these pores are larger than those of the voltage-gated ion channels in chemical synapses, meaning that a variety of substances can diffuse between the neurons. So now, how neurons communicate? We'll be talking about the resting potential. So the electrical signals move through the neuron as a result of changes in electrical charges of the axon. So normally, the axon remains in the resting potential. It is the state in which the interior of the neuron contains a greater number of negatively charged ions than does the area outside the cell. So now we're talking about the action potential. So when the segment of the axon that is closest to the cell body is stimulated by an electrical signals from the dendrites, and if this electrical signals is strong enough that it passes a certain level or threshold, the cell membrane in this first segment opens its gate, allowing positively charged sodium ion that were previously kept out to enter. And this change in electrical charge that occurs in a neuron when a nerve impulse is transmitted is known as the action potential. Once action potential occurs, the number of positive ions exceeds the number of negative ions in this segment, and the segment temporarily becomes positively charged. To illustrate, so alam natin, okay, so this is the axon, okay, at itong axon na to ay covered by the myelin sheath, and in between we have this node of Ranvier. Okay, so the myelin sheath and the nodes of Ranvier, wherein the axon is segmented by a series of breaks between the sausage-like segments of the myelin sheaths. Each of these gaps is the node of Ranvier. So the electrical charge moves down the axon from segment to segment in a set of small jumps, moving from one node to another node. When the action potential occurs in the first segment of the axon, it quickly creates a similar charge in the next segment, which then stimulates the next segment and so forth, as the positive electrical impulse continues all the way down to the end of the axon. As each new segment becomes positive, the membrane in the prior segment closes up again and the segment returns to its negative resting potential. In this way, the action potential is transmitted along the axon towards the terminal buttons. The entire response along the length of the axon is very fast, and it can happen up to 1,000 times each second. An important aspect of the action potential is that it operates in an all-or-nothing manner. What this means is that the neurons either fires completely, such that the action potential moves all the way down the axon, or it does not fire at all. Thus, neuron can provide more energy to the neuron down the line by firing faster, but not by firing more strongly. Furthermore, the neuron is prevented from repeated firing by the presence of a refractory period, which is a brief time after the firing of the axon in which the axon cannot fire again because the neuron has not yet returned to its resting potential. Okay. So for the anatomy and the structure of the synapse, so the purpose of this slide is to familiarize you with the anatomy of the synapse. So we have the vesicles. The vesicles are the packages containing neurotransmitters. Okay, so these are the vesicles. They must bind to the cell membrane and dispense their neurotransmitters through a process using exocytosis. Exocytosis is the process of having a synaptic vesicle containing various neurotransmitters fuse with the cell membrane and release the neurotransmitters into the synapse. Okay, so we have the axon terminal button. The part of the end of the axon that forms synapses with the postsynaptic dendrites, axons, or soma. So this is the axon terminal button. Then we have the presynaptic terminal button. So these are the presynaptic terminal button. It is the extension found at the end of the axon where information is conveyed to the next neuron. And this is the synaptic cleft or the synaptic gap. This is the small space separating the presynaptic and the postsynaptic cells. It is the space that neurotransmitter diffuses across to bind to receptors. These are neurotransmitters. These are chemical substances released by the presynaptic terminal button that act on the postsynaptic cell. And then these are the receptors. The binding site, this is the binding site for neurotransmitter. The neurotransmitter must fit in the receptor in a lock and key fashion. And when it binds, it opens an ion channel that allows ion to flow into or out of the postsynaptic cell. Then we have the spines. Spines are the protrusions on the dendrite of a neuron that form synapses with the terminal buttons of the presynaptic axon. Then we have, of course, the postsynaptic spine. It is a protrusion on the dendrite of an axon that forms synapses when the terminal buttons of the presynaptic cell. Not only do the neural signal travels via chemical or via electrical charges within the neuron, but they also travel via chemical transmission between neurons. So neurons are separated by junction areas, which is known as synapses. These are areas where the terminal buttons at the end of the axon of one neuron nearly but don't quite touch the dendrite of another neuron. The synapses provide a remarkable function because they allow each axon to communicate with many dendrites in neighboring cells. Because a neuron may have synaptic connection with thousands of other neurons, the communication link among the neurons in the nervous system allow for a highly sophisticated communication system. When the electrical impulse from the action potential reaches the end of the axon, it signals the terminal button to release the neurotransmitters into the synapse. Once again, the neurotransmitter is a chemical that relays signal across the synapses between neurons, and it travels across the synaptic spaces between the terminal button of one neuron and the dendrites of another neurons, where they bind to the dendrites in the neighboring neurons. Furthermore, different terminal buttons release different neurotransmitters, and different dendrites are particularly sensitive to different neurotransmitters. The dendrites will admit the neurotransmitters only if they are the right shape to fit in the receptor sites on the receiving neurons. And for this reason, as we said earlier, the receptor sites and the neurotransmitters are often compared to a lock and key. So when neurotransmitters are accepted by the receptors on the receiving neurons, their effect may either be excitatory, which means that they make the cell more likely to fire again, or inhibitory, which means they make the cell less likely to fire again. Furthermore, if the receiving neuron is able to accept more than one neurotransmitter, it will be influenced by the excitatory and inhibitory processes of each. If the excitatory effect of the neurotransmitters are greater than the inhibitory influences of the neurotransmitters, the neuron moves closer to its firing threshold. And if it reaches the threshold, the action potential and the process of transferring information through the neuron begins. So, after the neurotransmitters have been released into the synaptic cleft, they act upon postsynaptic receptors. Neurotransmitters that are not accepted by the receptor site must be removed from the synapse in order for the next potential stimulation of the neuron to happen. And this process occurs in part through the breaking down of neurotransmitters by an enzyme and in part through reuptake. Reuptake is a process in which neurotransmitters that are in the synapse are reabsorbed into the transmitting terminal buttons, ready to again be released after the neuron fire. Okay? So, sinasabi lang nun na lahat ng mga neurotransmitters na nandito sa synaptic gap na to, okay, will again go back dito sa pinanggalingan niyang end, okay, dito sa neuron na to. It will be reabsorbed or it will be disintegrated by a special enzyme. Okay? Pag bumalik na ulit yung neurotransmitter doon sa presynaptic neurons, okay, it will be ready again, sinasabi niya, for the next action potential to happen when the neurons fire again. Okay? So, there are several neurotransmitters, but for this particular slide, we will be discussing about some of the major neurotransmitters and their function. Because we know that there are more than 100 chemical substances which is produced in the body, which has been already identified as neurotransmitters. And these substances have a wide and profound effect on the emotion, the cognition, and behavior. And it regulates our appetite, our memory, our emotions, as well as our muscle actions and movement. Neurotransmitters are substances which neurons use to communicate with one another and with their target tissues in the process of synaptic transmission or the neurotransmission. Neurotransmitters are synthesized in and released from nerve endings into the synaptic cleft. From there, neurotransmitters bind to receptor proteins in the cellular membrane of the target tissues. And the target tissues get excited, inhibited, or functionally modified in some other way. Okay, so one of the major neurotransmitters are the acetylcholine. Okay, it is released from the motor neurons, basal ganglia, preganglionic neurons of the autonomic nervous system, postganglionic neurons of the presynaptic or parasympathetic nervous system, and the postganglionic neurons of the sympathetic nervous systems that innervates the sweat glands. And its major function is to regulate the sleep cycle, or it is essential for muscle functioning. Then we have the norepinephrine, also known as noradrenaline, which is released from brainstem, hypothalamus, and adrenal glands. And its functions was to increase the level of alertness and wakefulness. It stimulates various processes of the body. Then we have the epinephrine, also known as adrenaline. It's released from the chromaffin cells of the medulla of the adrenal gland, and the function is the fight or the flight response, okay, which is the increased heart rate, blood pressure, and glucose. Then we also have dopamine, which is released from the substantia nigra. It functions because it inhibits unnecessary movements, inhibits the release of prolactin, and stimulates the secretion of the growth hormone. Then we have the GABA, which is released from the neurons of the spinal cord, cerebellum, basal ganglia, and many areas of the cerebral cortex. And it reduces neuronal excitability throughout the nervous system. Then we have the glutamate, which is released from the sensory neurons and the cerebral cortex, and it regulates the central nervous system, excitability, learning process, and memory. Okay. We also have serotonin, released from the neurons of the brainstem and the gastrointestinal tract and thrombocytes, and it functions as to regulate body temperature, the perceptions of pain, emotions, and sleep cycle. Then we have histamine. Histamine is released from the hypothalamus, the cells of the stomach mucosa, mast cells, and basophils in the blood. And it regulates wakefulness, blood pressure, pain, sexual behavior. It increases the acidity of the stomach and mediates the inflammatory reactions. So for more information, I have included this link for additional. Okay, the psychoactive drug. Okay, corrects neurotransmitter imbalance. Okay, so we have the agonist, which mimics a neurotransmitter at the receptor site and thus strengthen its effect. Then we also have the antagonist. It blocks the normal activity of the neurotransmitters at the. Okay, so as a summary, we know that neurons, these are specialized cells which are found in the nervous system, which transmits informations, and it contains dendrite, a soma, and an axon. And some axons are covered with a fatty substance which is known as the myelin sheaths, which surrounds the axon, acting as the insulator, allowing faster transmission of electric. Then we have the dendrites. The dendrite is a tree-like extension that receives information from other neurons and transmit electrical stimulation to the soma. Then the axon. It is an elongated fiber that transfers information from the soma to the terminal button. Neurotransmitters. It relays information chemically from the terminal buttons and across the synapses to the receiving dendrites using lock and key type of system. And the many different neurotransmitters work together to influence cognition, memory, and behavior. Agonists. These are drugs that mimics the action of neurotransmitters, whereas antagonists are drugs that block the action of the neurotransmitter. Okay, so that's it for this particular chapter, and I'll be seeing you for the next chapter for the continuation of the biological perspective. So once again, thank you for listening, and I hope you subscribe and follow my YouTube channel. Thank you, and see you again. Bye.